Hello everyone, I am Professor Charu Dogra from Ramjas College, University of Delhi. I have been delivering lectures in the series Biotechnology, and today I will be talking about the molecular diagnosis of genetic diseases, in which I continue with the discussion of techniques, and today I will take up the techniques of PCR, SSCP and ASO dot plot method. The references used for this lecture is basically Glick, Pasternak and Patton for educational purposes, and some resources from the internet, some papers in which references are given in the particular slides. Now, molecular diagnosis, as we have been discussing, is the application of molecular biology tools and techniques to identify specific genetic mutations that are responsible for various hereditary diseases. Now, initially the identification or the diagnosis of these hereditary diseases used to be done on the basis of certain morphological changes or certain biochemical changes that could be assayed, and therefore the disease could be identified or diagnosed or predicted in the future generations. But all these techniques are basically very limited techniques. So, with the advent of various molecular biology tools and techniques, this diagnosis became very varied and more accurate, and things could be predicted in a very well manner. The prenatal diagnosis of Mendelian genetic disorders has been limited to conditions for which, as I said, the biochemical defect was known and expressed in fetal tissues such as the amniotic fluid cells, chorionic villi, blood, skin, liver. So, either the biochemical defect that could be assayed or certain phenotypic changes or morphological markers that could be identified. And for the prenatal diagnosis, the fetal tissues were used such as the amniotic fluid cells, chorionic villi, blood, skin, liver, etc. Of more than 2000 known Mendelian disorders, less than 100 are amenable to prenatal diagnosis using such kind of biochemical or morphological markers in fetal tissues. So, there was a lot of ethical concern as well, because the fetal tissues need to be excised and processed for these kind of markers. And second, there are very limited assays biochemically or morphologically that these diseases could exhibit and that could be diagnosed. So, very limited number of things could be, or diseases could be diagnosed. So, as I said, the identity of these mutations could be diagnosed very accurately by various molecular methods. The gold standard method to identify mutations is to directly sequencing the PCR amplicons. So, if we know and understand about the basis of these genetic disorders, like I would be generally taking up two examples, the cystic fibrosis and sickle cell anemia, and a somewhat beta thalassemia as well. So, these are arising by the single nucleotide mutations. And because of these mutations, the best and the gold standard method is to diagnose them by amplifying the certain genes which are responsible for the manifestation of diseases and simply sequencing them. So, once you know the entire sequence of the PCR amplicons of these genes, then you can compare it with the sequence of the normal gene, and you can identify the various mutations which are leading, or you can diagnose or detect the presence of these mutations that can lead to the identification of these diseases. Now, it is basically the simplest method. But problem is that it is a very expensive method, laborious method, and a time consuming method, particularly in large scale applications. So, when you have to identify a large number of samples, then sequencing, although we have discussed that the next generation sequencing techniques are evolving, and there are many techniques, and even in very less time they can give the entire sequence of the entire genome, and I am here talking about the PCR amplicons. So, obviously the things are improving at that end as well, but still there are certain molecular biology tools and techniques which are very simple, which are very handy, and that can be done to diagnose the various genetic diseases. diseases, and the sequencing, which is the ultimate thing that can reveal the identity of these mutations, but still it is a little expensive, laborious, or a time consuming method. So, in the last session we had discussed about the diagnosis of the diseases based on the RFLP method. Now, RFLP, just to briefly in one or two slides just review it. The RFLP is the restriction fragment length polymorphism based method in which the differences or the variations in the DNA sequence are identified at sites recognized by restriction enzymes. So, restriction enzymes or restriction endonucleases, we know, recognize certain recognition sequences and cut in those or near those recognition sequences. So, they generate different length fragments when the restriction endonuclease cut a genome. So, for example, if we are talking about a gene as well, then they may contain restriction enzyme sites for various enzymes. Now, when there is a mutation occur or when there is a variation or there is a difference in the DNA sequence, then it can alter the recognition site of that particular restriction enzyme. So, generally it can abruptly, when it creates a mutation, it can just render it inactive. So, there is no more recognition site that the restriction enzyme can recognize, or sometimes it can create a recognition site also. Most of the times it just abolishes the restriction enzyme site, but sometimes it can create as well. So, if it abolishes, then obviously if there is a particular gene and there is in the middle there is a restriction endonuclease site, and when it cuts it, it generates two fragments. But when there is a mutation in the recognition site and the recognition site abolishes, then when the restriction enzyme is used, it will not cut the DNA fragment and we will get a single sized fragment. So, such variation results in different sized DNA fragments produced by digesting the DNA with the restriction enzymes. So, very basic detection that we learnt about in the RFLP based method was basically the direct method, which was invented by Ballstein et al., in which the cells are isolated and from the cells the DNA is isolated, and then DNA is PCR amplified by using the primers which amplify that particular gene, and the fluorescent label is also used to tag these amplicons. Then these amplicons are digested with a particular restriction enzyme, and when it cuts the restriction enzyme, it gives different sized fragments which are then resolved on the agarose gel electrophoresis, and then the fluorescent label can be detected by the scanning machines. So, this is the basic detection method. Now, if a particular recognition site is abolished, as I was saying, then we will not get the same pattern which we will get in, for example, a normal individual. So, if the gene is mutated, then we will have a different pattern, and therefore different signal detection signal peaks would be detected that would reflect that there is a mutated gene that is present, and therefore we can predict that the disease can be there. So, this was a direct detection method. We also discussed about the indirect detection method, when there are no restriction or recognition sites in between the gene but adjacent to the gene, and with the help of that we can again, because of the linkage analysis, that particular restriction site which is nearer to the gene will be inherited in the same manner as the gene, and therefore it can also produce different sized fragments if it is abolished or created, and that indirect method can also be used where we do not have any recognition site for a restriction enzyme within a particular gene. So, these were the direct and indirect method which we discussed based upon the restriction fragment length polymorphism. Now, a related technique which I discussed today is the PCR SSCP. By Oreta et al. It was first used. Now, SSCP stands for the single strand conformation polymorphism. Now, as the name suggests, we will be looking at the single strand of the gene or the particular DNA, and then we will differentiate them based on the conformation. So, polymorphism again means that various kinds of conformation are there because of the single strand of DNA, because of the variation in the sequences. So, based on the initial separation or melting of the double stranded forms by heat into the single stranded form. So, the first the first step in this method is to basically separate the double stranded form of DNA. So, the initial separation, which is been done by heat, it is basically called as the melting, or it is generally called as the melting, whether done by heat or salts or anything. So, when the two strands separate, they will result in the formation of the single stranded forms. Now, we again know that these single stranded DNA can loop on itself or can form different conformational structures because of the occurrence of hairpin loops or the different kinds of double stranded structures that can be formed due to the presence of the complementary regions in a single stranded, just like the RNA forms that secondary structure. When the DNA is also double stranded, it can form different kinds of conformation, it can fold into different forms because of the presence of these different kinds of loops which are there. Now, in the separated state, the molecules tend to fold into a three-dimensional conformation according to their nucleic acid sequences. So, this concept is again very important, that according to the nucleic acid sequences, there might be certain regions which are complementary to each other, and therefore, they will fold onto itself because of the stem loop structures, hairpin loop structures, and all those kind of things. The hydrogen bonds can be formed between two regions of a single stranded structure and lead to a three-dimensional conformation. Now, when these single stranded forms are run on a polyacrylamide gel electrophoresis, a neutral polyacrylamide gel electrophoresis, then the size of the DNA strand is the same. Because it is the same gene that we are using, but still on the gel electrophoresis they are run at the different levels because of their different mobilities. So, again you can go back to the session on agarose gel electrophoresis and you can see when the DNA runs on the agarose gel, it runs based obviously under the influence of the electric field. It runs from the negative to the positive electrode, but the speed or the way it runs depends on the conformation of the DNA. So, if you remember in the double stranded forms also we discussed that the supercoiled formed will run faster than a linear or an open circular form. So, obviously there is a difference in the running on the gel electrophoresis. Running means the level at which they will reach, they will separate out even if they are of the same size, because the plasmid molecules were of the same size, but because of the conformations multiple bands used to appear. Similarly, the single strand DNA sequences for the gene that we are looking at would be the same in size, but will have different mobilities in the polyacrylamide gel electrophoresis, and thus it can be identified and diagnosed. Such structural conformations are affected by a mutation at a particular nucleotide position in a primary sequence after physical conformation of the denatured single strand bands. So, as I said that these structural conformations will be affected by a mutation. So, if a disease is causing any change in the base pair or any kind of mutations in that particular sequence, it will alter the physical conformation and therefore running on the gels, and therefore they will appear at the different bands. The alteration often leads to the slightly tilted positioning of the mutant single strand bands with respect to their normal counterparts in the neutral polyacrylamide gels. So, they will run at different levels, or their positioning will be little slightly tilted, or if they are recognized by, as I said, certain detection methods such as the fluorescent labels or the substrate enzyme complex label, then the peaks would be a little displaced. And therefore, we can identify whether it is the same sequence or the identical sequence of the gene, or there has been certain kind of a mutation that has occurred. So, this pictorial representation depicts basically the SSCP method that I have explained. So, for example, this is the DNA and this is the gene of interest we are talking about. So, this gene of interest we will amplify, and if there is a mutation and if there is not a mutation, we are looking at two different the two different a sequence of the genes. So, if there is no mutation, then as you can see, there are these base pairs which are there, and these base pairs, if you can see, when they are melted, when the two strands are separated, they are going to fold in a particular way. So, the AT or GC, they are folding in a particular way. But if there is a displacement or certain kind of a mutation which is there, where this base pair is altered in here, then when it folds, then you can see that the folding is different. The folding of these two molecules are the same, but the folding of these two molecules tagged with this blue fluorescent is different. And when they are run and detected on the scanner machine, then you can see that this peak for this particular nucleotide tends to appear a little displaced here. So, this because the conformation was same, although the nucleotide was changed, they are coming at the same point, but this particular nucleotide is displaced. So, it tells us that there is some kind of a mutation in this fragment because the tilt is displaced. So, the single strand conformation polymorphism can help in detecting the mutations and thus predicting or diagnosing the genetic disease that may occur. So, this is the pictorial representation. This is a characteristic SSCP. So, I have also taken a real example here. So, you can see that this is a particular gene for beta thalassemia, it's some 280 base pair gene, and then it is showing some 4 mutations which are there, these 1, 2, 3, 4 mutations in different regions. So, there is IS1 and IS2 and such regions are there, and in those regions you can see that there are certain mutations which are present here. So, when this singles, when this particular gene fragment can be PCR amplified, it can be done from the genomic DNA as well, but it is basically PCR amplified the gene of interest, and then it is made single stranded, and then it is allowed to fold itself, and when it fold itself, it gives certain conformations, and then it is run on the polyacrylamide gel or agarose gel electrophoresis, and then it results in the different bands which is there. So, if you look at the lanes, then this lower band is common, this last lane which is the lane 5 is the control. So, control means the normal individual, where there is no manifestation of the disease which is there, or we are not, we do not know that this is the person who is not suffering from beta thalassemia. So, it is going to give these two very closely associated bands here, and in all these organisms you can see, in all these samples you can see these two bands are present, and then there are certain additional bands which are present here. So, these are all heterozygotes. So, inheritance of the disease, we discussed it last time for cystic fibrosis and sickle cell anemia as well, that both of them were recessive genetic disorders. So, recessive genetic disorders mean that they will inherit one allele each from their parents, but they will express a disease only if the diseased allele is present in a homozygous recessive condition. So, homozygous dominant condition would not produce a disease, the heterozygotes will not produce a disease, but homozygous recessive condition would produce the disease of sickle cell anemia or cystic fibrosis as the case may be. So, all these are carrier or all these are homozygous, all these are heterozygous because they are having one normal allele as well. And in lane 1, 2, 3 and 4, you can see that there are different different kinds of mutations which are present here. So, for example, in lane 1, there is this ISI14 mutation which is present along with the normal. Then there is somewhere there is a codon 17 or codon 3 mutation which is present and along with the normal gene which is there. So, this is the normal gene, and these are the arising because of the differences in the conformations due to the differences in the sequence which has arisen from the different kinds of mutation. So, this is basically the PCR SSCP technique, and this is an example for a beta thalassemia gene that and they are all heterozygotes because they are giving one normal allele conformation of the single strand and one different alleles of the different conformations of the single strand where the mutation is there. So again, if we compare these two methods, the PCR RFLP method or PCR SSCP method, then there are certain limitations as well as there are certain advantages. So this is a PCR RFLP method. The one thing is that the PCR amplicons that can be analyzed by the PCR RFLP method is very much restricted. Why so? Because we require the restriction fragment length polymorphism, which certains that there is some kind of a restriction fragment length polymorphism which is there. So there has to be certain kind of restriction site or the recognition site which is present within the gene or nearer to the gene for doing a direct detection or indirect detection. So this makes this process very limiting, because all the genes would not be accompanied by a restriction site that can give rise to a polymorphism because of the certain mutation. So less amplicons can be studied here, and obviously the optimization is not although much required, because it can just run on the agarose gel electrophoresis, and therefore it is its recognition is very much limited. But if we consider SSCP, because no prior thing is needs to be known, in the single strand conformation, all you need is the DNA, the gene of interest amplicon, and you just need to melt it. So create the conformations under optimal conditions, they will fold into the different conformations. So a large number of amplicons can be analyzed by the SSCP method. However, their difference in their running on the gel requires lot of optimization. So the gels require lot of optimization. They have to be a denaturing, the neutral gels, so that it retains the conformation that the single strand fragments are going to take up, so that they can be differentiated on the gels when they are run. So obviously it requires lot of gel optimization, which is not present in RFLP cases, but in SSCP a lot of conformations are basically required, but a limited number of amplicons can be ascertained here, and a large number of amplicons can be studied here. So large diseases can be diagnosed by the SSCP method rather than the RFLP method. So, but again, they have much more, these are much more simple procedures, easy procedures, and this is a little cumbersome, not as cumbersome as sequencing, but a little, it requires optimization and all. So one needs to choose between like what method they use to adopt, the PCR RFLP method or the PCR SSCP method, based on their requirements and availability. So this is essentially the first method that I needed to discuss today, the PCR SSCP method. And in the RFLP method also, again, as I said, there are many advantages and disadvantages which are there, but the single strand conformation polymorphism relies only on the single strand sequence and the conformation that it attains. But here the major thing is the presence of the restriction site which is there. So not all the mutations carry the restriction sites that can result in the polymorphism, and thus it becomes much more limiting. So basically the different methods, the cystic fibrosis as well as the sickle cell anemia, or as I showed, the beta thalassemia gene, could be identified by both the methods, and one has to choose the diagnosis based on the requirement and the resources that they have. Thank you.